Now we will be joined by Ashley Zehnder, CEO and co-founder of Fauna Bio. Ashley is focused on the question, what if the key to unlocking human longevity was hidden in nature? Her team at Fauna is using hibernation, biology, and comparative genomics to develop novel therapeutics. Ashley is a veterinarian, scientist, and startup leader bridging biology and biotech in new and imaginative ways. Please welcome Dr. Ashley Zehnder.
Awesome. Thanks, Dylan. Yeah, I got it. Thanks to Dylan and Brenda for organizing a really, really exciting event. I will bring a slightly different perspective to how we think about longevity medicine. I will end up touching on some of the themes that we've been hearing throughout the morning in terms of the limitations of some of our existing animal models and also the value of repurposing pathways that we may already be looking at in new and interesting ways. But at Fauna Bio, as the title slide implies, we really look at animals that at the end of the day use the same genes that you and I have, but in very different ways, and in ways that allow them to not only resist, but actually in some cases reverse disease. And so it turns out that you can use genomics, you can use functional genomics, which is a fancy way of saying when genes are turned on and off, in a way that allow us to expand our understanding of health and disease across a number of different disease areas, many of which are diseases of aging. And so we think about this very much from including and increasing health span through the use of data and genomics. So we use AI largely to analyze large multidimensional data sets built from extreme animals, and I'll give an example of that, and merging that data with a lot of the same types of data that people are usually using for human health discovery, human genomics, and others. We do this in the near term to really think about what are drugs that we can put into clinical trials in the near future to impact diseases that affect millions of people around the world. But we're always thinking about how do the mechanisms and how do the pathways that we're examining link across diseases and help us ultimately live longer and healthier. We focus a lot at Fauna Bio, as Dylan mentioned, on hibernating mammals. My CSO and co-founder is not only a human geneticist, but a hibernation biologist. And so we focus a lot on the species called the 13-lined ground squirrel. So not a model organism you will typically see at a conference like this. People are used to working on things like mice and rats and worms and flies. This is what's called an emerging animal model. So there are species that are beyond that sphere of canonical model organisms that really have interesting biology that we can learn from and impact multiple diseases of aging. So in this case, this is an animal that hibernates for about 6 months out of the year. During that time, they drop their body temperature down to almost freezing. They go down to 4 degrees Celsius or about 40 degrees Fahrenheit. During that time, they are clinically brain dead. They have a flat EEG. Their heart rate goes from 250 beats a minute down to 5 beats per minute. Their bodies are 90% hypoxic or lacking of oxygen. And they stay in this period of essentially suspended animation for about 2 weeks at a time. But that is not actually what makes them the most interesting. What's most interesting about them is that every couple of weeks, they have to re-warm back up to normal body temperature in about an hour. So it's basically like going from being this frozen mammalian ice pack to being normal body temperature in an hour. It's a little bit like what happens after a human heart attack or a stroke. You get this rush of warm oxygen into blood into almost every major organ system in the body, including the heart and brain and others. This simulates pretty close to what happens after a human heart attack or stroke. You imagine if any of us had essentially a heart attack or stroke every 2 weeks over a 6-month period, we wouldn't be sitting in this conference room. So we can then study what happens as these tissues have to repair themselves from damage. We can also look at really dramatic changes that happen within the brain. These animals become hyper-tau phosphorylated pretty similarly to what happens in early Alzheimer's disease. This clears every time they go through a rewarming period. The neurons in their brain physically retract every time they go into torpor. So again, their brain waves are flat. They have flat EEG, clinically brain dead. They have to reconnect those neurons every time they rewarm. So they're a remarkable model for dendritic plasticity and the ability to retain long and short-term memory. They also have changes in terms of their oxygen status and how they handle changes in terms of collagen and inflammation in multiple organ systems, including the lung. We were talking about, someone asked about vision and maintaining vision. The cone photoreceptors in the back of the eye, they actually see color physically melt and reform every time they go through an interbout arousal. So they're a remarkable model for being able to restore vision. This is a 100-gram animal that's not moving for about 6 months out of the year. So you imagine a patient that's in the ICU for a surgery, is immobile for about 2 weeks, will start to lose a significant amount of muscle mass. These animals not only don't lose muscle mass, but they start to rebuild muscle toward the end of hibernation. So they're a remarkable model for regaining muscle mass that is lost for patients, for example, that are on GLP-1s. Also, when they're rewarming, they go from a metabolism about 1 to 3% of normal back up to normal body temperature. They increase about 235-fold their metabolism in a 1-hour period. My CSO likens it from going from a dead sleep to biking the Tour de France in about an hour, right? So it's really rapid rearousal. This is important for patients who have lost a significant amount of weight, but then their metabolism resets to a lower level. It's called the biggest loser effect. Makes it much harder for patients if they go off drug or if they go off their diet. They tend to rebound weight and sometimes will gain more than they've lost. Hence is why we've worked not only with Lilly but also with Novo Nordisk in the past to look for new therapies for obesity. And in the context of thinking about health policy, these are all diseases that affect a huge number of people, not only in the U.S. and globally, but just in the U.S., these diseases are responsible for over 1 million deaths every year, multiple millions of patients that have either geographic atrophy, which is related to the diseases we study here. Certainly muscle loss is an important indicator of aging in many people. And almost a third or a little over a third of adults in the U.S. are obese. And so these are diseases that affect really all of us or members of our families. This is just a quick little vignette to highlight some of the examples that I just showed you. On the histology, this is tissue histology, so looking essentially at a cross-section of the heart under the microscope. At the top is a normal human heart. In the middle is actually a human heart 1 to 3 hours after a heart attack. What's important to note here is really just that the fibers have become disorganized. There's also an increase in inflammation and collagen that goes on to lay down scar tissue. So in the bottom panel here, the light pink tissue is actually scar tissue, and that tissue becomes much less functional. So this is why the heart doesn't work as well after a heart attack. In the ground squirrel, what's interesting about these animals is not necessarily that they are protected, it's actually that they do get some damage. And so in this case, we have the top slide here. Normal squirrel heart histology looks very similar to our own. But after, when they're rewarming during hibernation, as I mentioned, at the top of those intervital arousals, they have similar kinds of damage. You see this wavy fiber disorganization. You see changes in inflammation and collagen. But at the bottom, we see a heart that is fully repaired, having had this damage happen, I said, 25 times over a 6-month period. So it's a really remarkable model for natural tissue healing. And it's molecules that, pathways that have really been retained across hundreds of millions of years of evolution. And so we'll talk a little bit about that. We can look directly at tissue repair, but these are animals that, because they have to survive really extreme environments and really extreme changes to their physiology, have optimized many different levels of what we call hallmarks of aging. And obviously there's been a lot of talk about that at this conference, but I'll just highlight that for the fact that we can then use these models to look across multiple different of these pathways. We mostly focus at Fauna on these direct pathways of cellular regeneration, cellular healing, reduction of cellular stress and mitochondrial function, which is actually the lead program that we have works on all these mechanisms. And so again, you can look at many of these lenses through the lens of hibernation and affect these interrelated pathways. We do collaborate with large government-backed institutions like the Broad Institute, who funded a work called Zoonomia. This was an effort to look at genomic conservation across really all of the sequenced mammals. It started out as a project called the 29 Mammals Project. It's now called Zoonomia because now it's up to 452 mammals. The goal of this was to be able to align mammalian genomes and actually look and see which parts of the genome haven't changed in 100 million years. With the thought behind this being very simply, if something hasn't changed in 100 million years, it's probably doing something important. And so then what are those important genes doing in the context of disease? What's interesting about hibernation as a lens for this type of analysis is that there are actually hibernators across almost every major group of mammals. The blank spot over here in the corner is hoofstock and marine mammals, but every other group has hibernators, including primates. So there are lemurs in Madagascar that hibernate very similarly to ground squirrels and have similar kinds of protection. And so for us, it's really a lens into ancient conserved pathways of tissue repair and healing that these animals have maintained and that we have lost, and that now we're able to think about reactivating through the therapies that we derive. This is just showing some of the lifespan of some of these creatures. So a ground squirrel, for example, that we work on, normally a 100-gram animal, which is a little bit bigger than your average lab mouse, about the size of your average lab rat, which lives about 2 or 3 years. These animals live closer to 8 or 9 years. If you look across all of these species, those that hibernate do have a longevity advantage, even if you account for the metabolic cost savings that they have during hibernation. And so then the question becomes, well, this is all very cool, but how do we actually translate this into insights that will help people at the end of the day? So this is where the insights for my other co-founder, Linda Goodman, and my CTO comes in. She has a background in human genetics, but also computational and evolutionary genomics. And so we built a platform called Convergence that does as the name implies. It takes data from these extremely adapted animals, particularly hibernation, but we can work with other species as well. Intersects that with human data, human genomics, and human functional data, allows us then to map onto conservation data, so how conserved are genes and pathways across all the mammals that we have studied. We then take genes and pathways that we've identified through this approach, we then test them on the bench in human cell lines. So can we protect human cardiac cells from damage? Can we protect human neurons from damage? Can we protect function in human retinal cells? And really what we're looking for is this pattern of opposite versus damage signatures. So can we see a pattern of protection in a species like a ground squirrel? Can we literally map that onto genes that are turning on or off in, say, humans with heart failure? And can we start to enrich for pathways where we see protection in a species like a ground squirrel that's dysregulated in human diseases? We then can map that directly onto compound libraries. So we actually say, are there drugs that exist now that if you treat human cells with those drugs, we can mimic these natural protection signatures, and it gives us a jump start into getting drugs into the clinic to help patients faster. And so this is a really quick vignette of how we actually got to our current lead program, Fauna 1083. This is a pattern of genetic signatures here on the left. The boxes here are genes that are either going up or down or expression, or essentially turning on or turning off. The time points across the bottom are all hibernation time points. And so we're looking across the hibernation cycle to find genes that are turning on when we know these cycles of damage are happening. We then can map that to chemical libraries. This is another government-funded effort. This is from NIH. It's a library called Links 1000 data set. It allows us to map gene expressions to chemistry. And that allowed us to find a tool compound, Fauna 1003, that we then took through a lead optimization process to find what is now our development candidate, Fauna 1083, which we're hoping to put in the clinic in a little bit less than a year. This is for a disease called heart failure with preserved ejection fraction. This is an not emerging, but increasing in prevalence type of heart failure, different than our sort of traditional type of heart failure, which is reduced ejection fraction. It's important because this is a type of heart failure that really increases both with aging and obesity, and due to a number of different systemic risk factors like diabetes and hypertension. So a lot of the risk factors that we've been talking about. Over time, as these hearts become less functional, blood backs up essentially into the pulmonary vasculature, and you have pulmonary hypertension as well. There's around 3 million patients in the U.S. with this type of heart failure. Again, this is increasing with age, also is more prevalent in women, and about 50% of those patients will also have pulmonary hypertension. And so our drug 1083 works on both mitochondrial energetics and also cycles of inflammation that happen to improve outcomes in animal models. We do work with traditional animal models as well. We just don't use them for discovery, but we do use them for confirmation. Here we're using a genetic model for this type of heart failure. It combines a diabetic rat model with a lean hypertensive model. It develops a rat called a ZSF1 rat that actually has multiple different backgrounds that are similar to what happens in patients with this type of heart failure, namely obesity, insulin resistance, hypertension. And they develop the similar kinds of exercise intolerance and muscle loss and vascular function impairment that happen in patients with HFpEF. This is my one data slide in this talk, but this is a marker called E/E prime ratio. Really, it's just a measure of how well the ventricle can relax and allow blood to fill, which is the primary dysfunction in this disease. What you want to see is this number staying low. This is what happens in our controls. What we see in the blue line here is our untreated obese animals. This stays very high, whereas in our treated obese animals, this number gets much more closer to normal. That's important because as the blood is able to fill the heart, these animals are able to then exercise better, and that's a measure that we're working on right now. But being able to improve a marker called the pulmonary capillary wedge pressure, this is a good predictor of that. That's actually a good measure, a proxy for 6-minute walk test, which ends up being a clinical outcome measure for heart failure. So it's an early indicator that we're having a functional effect that should be able to improve exercise tolerance at the end of the day in healthy patients, and will help us improve lives in patients. Here, this is just comparing to, I know other people have mentioned looking at an empagliflozin or Jardiance. These are SGLT2 inhibitors, these are already mentioned today. We just controlled, published studies in the same model looking at improvements in E/E prime ratio, showing that we have a better impact more quickly than the approved drugs for this indication, which are Jardiance and Entresto. And this makes sense for us because we're treating actually a target that's expressed in the heart muscle cells and having a direct effect on the heart's ability to adapt to inflammation and loss of mitochondrial function. And so I'll just briefly highlight here, this is not the only thing that we're working on. I mentioned we had the discovery program with Eli Lilly for obesity. We're working in the retinal space as well, and we do have some earlier discovery programs in the neurologic space. We do have active collaborations with multiple academic labs. This is really, really important for our work, this academic industry collaboration. There are oftentimes maybe only 1 or 2 labs in the world that work with these species, and so we work directly with those labs to get data from species that have these really innovative approach to disease modulation. We are funded also directly through the government. We've had previous STTR and SBIR grants support our earlier work, and we're currently funded on 2 of our programs through NASA for implications on hibernation biology and space technology. So what do we really need to support this kind of really innovative approach to aging biology? As I mentioned, just supporting early stage research in emerging animal models. These animal models are often considered quite niche in biomedical research. They often fall in funding cracks between NSF and NIH. NSF considers them too translational, NIH considers them not translational enough, and we're scrambling in the middle. The 13-line ground squirrel is awesome, but they are not the only very cool animal that is emerging that we can use for our work. There are over 35 other species that have unique types of disease resistance that we could start to study. And you'll notice if you look at the types of diseases here, it's everything from cancer to muscle atrophy to aging that are just kind of waiting to be discovered. So regulatory support, as I mentioned, having a clear path for looking at endpoints across diseases and diseases of aging is really important. Supporting and having robust architecture for building academic industry collaborations. There is work that is more appropriate for academic labs, like setting up these models. There's work that's more appropriate for industry, like translating them into therapies. You need both for that to happen. And continuing support for early stage biotech, having lean and efficient methods to support early stage biotech. Like I said, we had SBIR and STTR grants. The administrative overhead on those things can be quite burdensome for small biotech, so finding lean, efficient ways to fund early biotech work, super, super, super important for biotech. And then with that, I'll take any questions. Thank you.
One question.
Behind you.
Really excellent presentation. I was wondering, if you subtract the amount of time actually spent hibernating, is there still a differential in lifespan?
Yeah, that was a question. It's hard, right? Because you have a reduced predation, you have reduced metabolism, and so roughly, if you try to account for that, there have been studies that look sort of across those mammals, and it's sort of between a 20 to 30% survival advantage, even if you account for that. What we're more interested in are sort of these direct tissue repair pathways, but yes, it seems like there is some survival advantage. It's not doubling or anything, but again, these are animals that are also hidden from predators and have a low metabolism for about half the year. Yeah.
Awesome, thanks.
Yep.